Assalamualaikum warahmatullahi wabarakatuh. Good morning, Shalom Om Swastiastu, Namo Buddhaya, Salam Kebajikan. Welcome to the international webinar Pre-APFITA Series Chapter 3 with the theme Plan, Pandemic, Current Status and Future Perspective. Allow me, Nanda Ibrani, to guide today's event as a master of ceremony. The Honorable Professor Dr. Insinyur Yandra Arkaman, Master of Engineering, Head of Logistic and Agro-Maritime Study Program, Chair of Brain IPB University, and President of APFITA 2025. The Honorable Professor Seishi Ninomiya, PhD, from the Phenomics Laboratory, Graduate School of Agriculture and Life Science, University of Tokyo as our speaker. The Honorable Professor Dr. Engineering Wisnu Ananta Kusuma, STMT from the Department of Computer Science, Faculty of Mathematics and Life Science IPB University as speaker. The Honorable Mr. Verry Surya Hendrawan, lecturer at the Faculty of Science and Technology Muhammadiyah University of Cileungsi as moderator. And ladies and gentlemen, as a wonderful webinar participant. Ladies and gentlemen, did you know about APFITA? Let me quickly introduce about APFITA. This event is a part of our successful APFITA seminar series, which has been held 3 times, and the next session will take place in November. And then I will tell you about the rules webinar today. Webinar rules today. The first rules, participant must use a Zoom with the format name_affiliation. The second rules, mute your microphone during the session. And the last rules, question can be submitted directly by sending a direct message to the committee with the name Rian_question and Kania_question. Before we move on the next station, let us take our proper and respectful position to sing the national anthem Indonesia Raya and continue with hymne IPB. Thank you. To begin this event, we respectfully invite Professor Dr. Insinyur Yandra Arkaman, Master of Engineering, to deliver the opening speech and officially open the webinar.
Baik, terima kasih. Morning all. Assalamualaikum warahmatullahi wabarakatuh. Good morning. Distinguished guests, speaker,
We cannot hear Prof. Yandra, Mbak
Hello,
MC.
hello, good morning. Good morning.
Okay, yeah, please closer to the microphone.
Yeah, good morning, distinguished guests, esteemed speakers, Professor Ninomiya, and fellow participants from across academia, from everywhere, the world, industry, government, and general public. It's with great enthusiasm that we welcome you to the National Webinar Series 12, Pre-APFITA Series 3, centered on the cutting edge of plant phenomics. Our topic today is about plant phenomics. Plant phenomics open a new frontier in agricultural research, especially also for IPB University. One of our focus for three next 3 year research is about the plant phenomics. Over the course of today's session, we will hear from both renowned experts who will share groundbreaking insight into sensing technologies, data acquisition challenges, and transformative power of AI and high performance computing in genomic integration. Also, did one research with Professor Wisnu before, Professor Ninomiya, of course, we will share a topic, valuable material for us today. We hope this webinar inspires collaboration, sparks innovative ideas, and empowers all of us to contribute to the future of sustainable agriculture and smart farming system. Thank you for joining us on this exciting journey of discovery and progress. Have a wonderful conference in webinar APFITA today. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Professor Dr. Insinyur Yandra Arkaman, Master of Engineering, for your opening remarks. Next, we will take a group photo. We kindly ask all attendees, ladies and gentlemen, who are available to turn on your camera for documentation for this morning. Please assist the committee for help this. So guys, please, ladies and gentlemen, to turn on your camera for documentation. One, turn on your camera, guys, please. 1, 2, 3. One again. Stop. Okay, repeat again. 1, 2, 3. Give your best style. The last. 1, 2, 3. Okay. Now, it's time for us to move the main session. To guide the presentation and discussion, we have Mr. Verry Hendrawan, lecturer in the Faculty of Science and Technology Muhammadiyah University of Cileungsi. Mr. Verry Hendrawan have amazing experience, professional experience like researchers brain APB University, lectures, commissioners, assist to the presidential special staff, member drafting team presidential, and member Halal Product Fatwa Committee, and have formal education bachelor degree in mechanical engineering, master degree in management, doctoral degree ongoing in agro-industrial technology APB University, and work have experience over 20 years of experience in manufacturing industry. To Mr. Verry Hendrawan, please lead the presentation and discussion session.
Thank you so much, Mbak Nanda, the MC. Ladies and gentlemen, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning to everybody. Thank you for the opportunity given to me for moderating this very interesting webinar. But before we start, allow me to share a little bit about APFITA, as what mentioned earlier by the MC. As you can see on the screen, APFITA stands for the Asia Pacific Federation for Information Technology in Agriculture. It's an international federation dedicated to promote the advancement and application of information technology, digital innovation, and smart solution in agriculture, fisheries, and agro-maritime sector across the Asia Pacific region. This year is going to be the 15th edition of this prestigious global scientific forum. The 15th International Conference of APFITA is proudly hosted by IPB University in Bogor. This international conference will be held on November 17 to 19, 2025, located in IPB Convention Center, Bogor, Indonesia. Please, let us join with all total delegates from 15 countries, including world-renowned researchers, academics, policymakers, practitioners, to discuss the latest innovation in digital agriculture and sustainable agro-maritime development. For more information, please feel free to check on the website as presented on the screen. All right, shall we move to the session that we have been waiting for, which is the webinar? Go to the next slide, please. Next, please. Okay, so now we are going to have the first presenter, which is who is Professor Seishi Ninomiya. But before that, I would like to warm remarks to distinguished professor, Prof. Seishi Ninomiya, Prof. Wisnu Ananta Kusuma, and Prof. Yandra. Today, we are going to explore one of the most exciting frontiers in agricultural science, which is plant phenomics, a discipline that aims to comprehensively measure and analyze plant phenotypes to understand the complex interaction between genotype, phenotypes, and environment. And with the rapid advancement of multispectral imaging, high-speed sensor, robotics, and artificial intelligence, plant phenomics has become a key enabler for, first, accelerating plant breeding efficiency, secondly, improving precision cultivation management, and third, reducing environmental impact through data-driven decision making. We are honored today to have two distinguished speakers joining us, Professor Seishi Ninomiya from Tokyo University and Professor Wisnu Ananta Kusuma from IPB University. So without further ado, let us begin our first session. But before that, please allow me to share an impressive resume of Prof. Seishi Ninomiya. His research field is on plant breeding and physiology, applied science, statistics and agriculture, computer science application in agriculture, and plant phenomics and digital agriculture. With all a long story of academic and professional position, he is a professor emeritus, University of Tokyo, Japan, Professor Nanjing Agricultural University, China, past president of SciGR, editor-in-chief of Plant Phenomics Journal, member Agriculture, Forestry and Fisheries Technology Council, Ministry of Agriculture, Forestry and Fisheries of Japan, research director, National Agriculture and Food Research Organization, NARO, Japan. And as all we can see in the screen, his research leadership experience, award and honor, and professional recognition all add up together in his impressive resume. Again, without further ado, allow me to deliver this stage to Professor Ninomiya. Prof, the floor is yours. You have 30 minutes to deliver your presentation. Please, Professor.
Okay, can you hear me? Hello?
Very loud and clear. Yes, Prof. Please.
Yeah, okay, thank you very much for the detailed explanation. And I'm very glad to have this opportunity to talk about our studies on the plant phenomics. Well, let me share screen. Does it look okay?
Yes, we can see your screen, Prof. Please.
Okay, very good. And last night, I have counted how many times I have been to IPB before, and I finished the counting after the number reached 15. So at least I've been there more than 15 times. I'm very glad looking forward to attending the next conference in Bogor in November. And today's title is Plant Phenomics. Moment. Okay, so the phenomics is an interdisciplinary science field for phenotyping. It's trait evaluation. It's phenomics versus genomics. It's a kind of anonyms of genomics. Okay, and actually, this graph shows the number of the papers published in last 35 years. You see that the last 10 years, that this field it was developed so quickly and now reaching a kind of a plateau time. And my first study for plant phenomics was done almost 30, 40 years ago. This is one of the studies we have done about the replacement of the visual judgment of the breeders. You see that in the breeding process, sometimes the breeders made a selection based on the shape of the crops. This is the case for soybean. And I wanted to replace the visual judgment by the breeders by image analysis. And I don't go to the detail, but finally we realized that this kind of image approach can simulate the human visual judgment very accurately. The accuracy was almost 95%, or even higher than the other one. So we really got convinced with that this approach would be very, very useful. This is a very, very fast starting point of the plant phenomics. Now, I belong to the University Tokyo, and my office located in the University Tokyo farm, and which is not very far from the central Tokyo, but still we are keeping about 40 hectare land there. And we have a quite interdisciplinary team for plant phenomics, because the plant phenomics need a lot of different types of the field of sciences, including breeding science, crop science, and informatics, engineering, etc. This is a really true interdisciplinary field of science. And we mainly focuses on the outdoor field phenotyping, not indoor things, and which is more difficult than the phenotyping indoor. But anyway, we've been doing quite a lot of studies on this. And we really believe that the phenomics takes a vital role in achieving sustainable and productive agriculture. And the objectives are the first step is the quantification of trait. I will talk about this later on. And the second thing is the replacement of inefficient human action to high throughput ways in space and time in phenotyping, and also the visualization of the trait with the new sensors. With this new approach, it can benefit a lot. Which can provide a very high efficiency of data collection for big data generation in AI era, which is necessary in AI era, and also which can provide totally new traits that a human hardly observe, such as frequent time series observations or spatial observations, etc. And also nowadays, the 3D structure construction became possible, which is also used to be almost impossible before, say just 10 years ago. And also so-called functional traits, such as water use efficiency, radiation use efficiency, photosynthesis, etc., became available. And now we can apply this new approach as a monitoring tool in a smart farming, because this approach can very effectively replace the farmer's visual judgment. And the second one, which might be the largest benefit of the high throughput phenotyping, is to accelerate the breeding process. And through the studies of high throughput phenotyping, we are realizing that the high throughput phenotyping can even accelerate the new discovery, new biological or plant science discoveries in the last 10 years. Now let's go back to the original question. So the first thing which is brought by the new phenomics is that the quantification of trait. You see, this is a book about how to evaluate genetic resources in Japan, and it is a case for the shape of the potato. But you see that all the traits, like a shape, is described very qualitatively, which means that we cannot make as a data. We cannot use them as data. So the very first step of this field was the quantification. You see on left-hand side, this is again case for the leaf shape of the soybean. And I don't think I have enough time to explain the things in detail, but anyway, I kind of using a so-called Fourier descriptors, we realized that 80 descriptors can describe the leaf shape of the soybean very precisely, accurately, but because 80 variables are too many, so we decided to get the principal component to reduce the number of the variables, and we realized that only 5 principal components can describe the shape of the soybean very accurately. Based on this approach, we succeeded in finding out the shape genes which control the shape of the soybean, you see. And also look at the left top. This is a flower color pattern of the pansy flower. But even that kind of things can be now quantified. And the right bottom, this is a barley kernels. And you see the left kernel doesn't have any wrinkles, but on the right one has a lot of wrinkles. This wrinkleness is very important characteristics for malt quality. Malt is I don't think you produce a lot in Indonesia, but the malt is used for the whiskey and the beer production. And this wrinkleness is very important characteristics which determines the quality of the malt. So also we succeeded in quantifying this kind of very complicated shape structure as well. And also as I mentioned, the one important factor of the current phenomics is a kind of replacing the human actions to kind of fully automated and high performance high performance observations using a kind of new sensors. And another thing is I mentioned that acceleration, the breeding can be provided by the phenomics. And you see the ultimate goal of in this aspect of the phenomics is to make a model to explain a phenotype by genotype, environmental condition, and management. You see, so we believe that the phenotype can be determined by 3 factors, three conditions. So once we have this model, we can accelerate. We can understand the function of the crops or plants, how they're correlated, how they have a kind of a relationship among those factors, and which mean that we can accelerate the breeding, for example, because in the breeding process on the left, you see after crossing, the breeders have to conduct the selections every year. They have to grow for example, rice in the paddy every year, and they make observation once a year. So for example, in the case of the rice, we need maybe 10 years to develop just one cultivar. But in once we have this model, what we have to do is to accelerate the generation, the reproduction. So in only 1 year, maybe we can have the 4 productions. We can get 4 cycles of the seedings, which means that we can accelerate because we don't have to conduct the selection in the field. Because using this model, we can predict the phenotype from the model. So what we need is to collect just seed. Then we have using these seeds, we analyze the DNA and the genomic information. Then based on the genomic information, we can predict the phenotypes. This can accelerate the breeding process very, very efficiently. Another thing that I mentioned that the Phenomics can help the smart farming as well. On the left, in farming, we have 3 steps. First, we make an observation in a field, crop status, environmental condition, etc. And based on the observation, the farmers make decisions. And based on the decisions, farmers conduct some management in the field. They repeat this cycle. And in conventional farming, in the orange one, everything is done, empirically, based on the human judgment. But on smart farming, every step is done fully automatically, for example, based on some images, etc. So the Phenomics works very effectively in those 3 steps in a smart farming as well, replacing the human visual judgment. Now we have a lot of sensors, and I don't think I'm reading one by one, but visual light urging sensors, multispectral imaging sensors, hyperspectral imaging, thermal infrared imaging, chlorophyll fluorescence sensors, and also there are several different 3D reconstruction sensors and approaches. Okay, for example, okay, now on the left top, it is quite easy to detect green tomatoes under the green leaves. Just 10 years ago, it was almost impossible. On the left bottom, you see this kind of 3D reconstruction of the orchard is quite easy nowadays using UAV images. And on the left shows the whole year growth of soybean breeding field. You see each of the plots shows a different growth patterns depending on the genotypes. And now this kind of 3D reconstruction becomes quite possible if you fly UAV every 3 days in a field, for example. Now, another thing which is brought by high fruit phenotyping is new traits, which were almost impossible observable by human approaches. One of the typical example is of the time series dataset. Okay, this shows again the case for the soybean breeding field, and on the right top shows the change of the height of each of the cultivars we are growing in the field. And now using a 3D reconstruction approach, we can have the height of the field of every plot very easily based on the images collected by UAV. And also the same thing can take place with the canopy coverage. And you see that it was almost impossible for human to do this before. But now it is very easy. And now we are understanding that this time series curve itself is a new trait because they are different among different genotypes. And we just realizing that the different curve can strongly influences the yield performance as well. The other example is the detection of the crop organs. On the left, yeah, we succeeded in detecting the flowering of the rice panicles. Rice panicle, you see, it's a very small, happening in the field. But now we can count number of the panicles flowering in a field on time series. And let's see, you can see the kind of diurnal change of the flowering in the field. And they are quite different among the different cultivars. And also, we can track each individual panicle from the beginning to the end, which means that we can see the performance of each of the panicles, and we realize that this flowering, I mean, we can track each of the panicle. Then we realize that this flowering timing of the panicle is influenced by the fertilizer levels. Another example is the soybean seed counting in the field. That we don't need to take them into the indoor, but we can count the number of the seed in pods of the soybean in the field, so that we can have. Please look at the right. We can have the seed number distribution of different cultivars. And this characteristics very important for design the robot harvester. Another thing is growth uniformity. We realized that depending on the wheat cultivars, I mean uniformity in each of the crop was different. And also we realized that this uniformity also influences in the performance of the wheat as well. So these are all new traits which were not available only with the human observations. Another example is the identification of annual and pruned branches of this is for pear tree. And you see, by 3D reconstructing the peach tree with before pruning and after pruning, and after pruning and before pruning, we can identify which are the annual new branches, which branches were pruned, etc. And now we can have this kind of distribution about the angle distribution of annual and the pruned angles, or length distribution of annual and pruned branches, shoots. Okay, this is totally new information. But now the breeders realizing that this information may be important for the productivity or the quality of the pear tree, pear fruits. And just I spoke about some of the element technology in the plant phenotyping, but something most important is how to integrate those technologies all together to make those technology applicable to the real field. I show you one example here, and this is a case for a broccoli, and I mean the broccoli ball do not equally uniformly grow in a field. For example, in this field there are more than 10,000 broccoli, but the sides are different. So and also I have to mention that the price of the ball is different. Large one between 12 to 13 centimeter is most expensive, then the middle one, etc. So the farmers has to identify most optimal timing of the harvest if they have to harvest all the broccolis all together, you see. So this is not an easy thing, because they don't have to, they cannot distribution of the broccoli size. It's almost impossible for them to measure the size distribution on a field. So what we took, yes, okay, just on the left, yeah, we have to have we have to know that size distribution like this to maximize the price of the I mean maximize the benefits. So we need three technologies. One is each ball detection, identification. We should not miss any, even one broccoli. We have to detect everything, even though they are partially occluded by the leaves. Then second step is to estimate the size of the broccoli, even though those balls are partially occluded. Then the next step is to predict the growth of the broccoli size in coming 2 weeks. So combining 3 broccolis, we can estimate the size distribution. I mean, we can know the growth of every broccoli in the field, even though there are 10,000 broccolis in the field. Then we can have the distribution of the incoming 2 weeks, so that finally we can have the most, we can identify the most optimal date of the harvest, in which the loss of the benefit is minimized. Okay. So now data is a key driver of the AI era. I have shown you some of the examples of the new phenotyping technologies. That most of them use AI approaches. And so to have a big data is most important in this study. And this is a case for the wheat data set, and more than 10 different institutions of the different countries just agreed to combine all the data sets they have, making their wheat data set so huge. And this approach is really good. And all the models based on this data was much better than independent models provided by each of the institute. And one of the fruits by this collaboration is that weight foundation model, which can be applicable to several different purposes, such as leaf counting, organ detection, organ segmentation, etc. Another thing I have to emphasize that utilization of historical data, because in agriculture we usually can have only one crop. Of course, sometimes we can have 3 crops, 4 crops in the case of rice, but usually we can have one crop, which means that to have the 20 replications of the same plot, we have to wait for another 20 years. This is kind of, we cannot wait for that long. So only the way we can do is to go back to history to collect data. And now we have started to digitizing all the historical data to enrich our data set, to make it available for AI approaches. So this is one thing. Now, I would say that the data orthogonality or normality and the format, etc., not really necessary in this area. We used to worry about these things, including standardization, but it is not really highly required. And for example, just I took a photo from some presentation. It is quite easy to make a digitized version of dataset. Everything is quite much easier than before. So now we have a lot of tools to digitize some kind of available dataset. And another thing we have to emphasize is the interoperable data platform. Yeah, we do, after having a data set, we do have to provide some interoperable data platform to maximize the data usage. This is a case of WAGRI, which is provided by a data platform provided by NARO, National Agriculture and Food Research Organization of Japan, and now it's becoming operational to combine several different data sets. And now the performance of developing new data set and new applications are much better than before based on this approach. And now this is probably the last slide of my presentation. And still there are lots of challenges in the plant phenomics. Foundation model is a kind of AI model for generic model, because to make this one, we need more data. And I talked about the 3D modelings or 3D reconstruction. but still a lot of problems. And root phenotyping, yes, this is one of the most difficult one because at this moment, people are using CT or X-ray or something else. So no way to do this in a field efficiently, etc. Lots of issues. And now I think this is the last slide. And yeah, I think the time has come. So I don't like to read the things one by one. But for our research, we have a lot of collaborators internationally, domestically, and lots of private sectors, etc., doing this research. And for the final one, yes, we are very glad that we are approved that by the organizer of APFITA 2025 to organize a special session for a plant phenotyping. And we are planning to launch a new group named the Pan-Asia Pacific Plant Phenotypic Network, collaborating with the IPPN, International Plant Phenotypic Network, which is a quite huge group internationally. Yeah, there is one in the America, one in the Europe, etc., but not in Asia. Yeah, in some of the countries, for example, we have a JPPN, and in Korea, KPPN, etc., but still we don't have any Asia Pacific organization. So we are planning to launch a new network for this new field of interdisciplinary science. Okay, thank you very much. And the presentation I have shown you is available from QR code. Okay, so please, I will keep this presentation till the maybe end of this month. So if you get a photo of this QR code, you can download the almost the same presentation as I have talked about. Okay, thank you very much for listening.
Thank you very much, Professor Seishi Ninomiya. Arigato gozaimashita. This is so insightful and inspiring presentation. It's just so amazing to see on how the digital advanced technology has been supporting for the fast development on these particular sectors. On how, for example, to how do they trace, to count, to deliver a better vision, and so on and so forth. We truly appreciate for your comprehensive explanation on how plant phenomics is evolving and shaping the future of the crop science. Please, Professor Seishi Ninomiya, keep stay on the room. We will be having the second center and then we will be having the Q&A session. Please stay, Prof.
Okay.
So respected researchers, lecturers, students, and all participants joining us from various institutions across Indonesia and abroad, it's again my great pleasure to welcome you all to today's international webinars on plant phenomics and current status and future perspective. We've been through the first presenter for Professor Seishi Ninomiya. Now let us move to our second speaker, Professor Wisnu Ananta Kusuma, who will discuss the opportunities and challenges of applying plant phenomics in tropical environments and its contributions to Indonesia food security and smart agriculture. But before that, please allow me to share his impressive resume as well. Could you please go to the next slide? Yes, Professor Wisnu Ananta Kusuma. His formal education from ITB, graduate, postgraduate, and the doctoral degree from, I think it's not Tokyo Institute, but it's in Haneda, right, Professor Wisnu? Correct me if I'm wrong.
Tokyo
Sorry
Institute of Technology. Yes, it's correct.
Oh, that's correct.
Tokyo Institute.
All right. Thank you. Thank you
But,
so much. Thank you so much, Prof. Okay, and as you all can see, he has so many awards and honor. Of course, if we put everything here, is not be able to manage or to cope all of them. But let me show you some. 107 innovation in Indonesia. Satya Lencana, tribute to innovator in IPB University, Best Paper Award of International Conference on Bioinformatics, Biotechnology and Biomedical Engineering, Biomic 2020, Best Paper Award of International Conference on Bioinformatics, Biotechnology and Biomedical, Biomics 2021. Diamond Award category in IT for health. His research focus in bioinformatics and computational biology, AI and machine learning, natural product and drug discovery, network pharmacology and molecular docking, as well as high performance computing. And he has been invited to so many sessions in both seminar and webinar. So without further ado, let's now go to the next speaker, who is Professor Wisnu Ananta Kusuma. Professor Wisnu, please proceed, Prof. You have 20 minutes, but of course, we can have more time if needed. Go.
But I cannot share. Still
Oh,
not have
you cannot share?
access. Yes.
Okay. Please give access, team, panitia, committee.
Okay. nih.
All right, Prof. We can see your presentation. We can hear you clearly. Go ahead.
Okay. Yes. Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Firstly, I would like to thank Professor Yandra and also the committee that inviting me to have a opportunity to give a presentation in this webinar. Because I'm a computer science, so I'm a computer scientist, so I would like to focus on machine learning approaches to genome-wide association studies. I just want to introduce myself. I'm a lecturer in computer science School of Data Science, Mathematics, and Informatics, and also the researchers in tropical biopharmaca research centers. Actually, after graduated from my doctoral degree, I start research in the topic of agriculture, especially how to use the genomics, omics data or omics technology to improve readings based on the genomics and phenomics associations. But after maybe from the 2018, I moved to the topics focus on the health systems and drug discovery. So in this presentation, maybe I will show our results, especially in the GWAS study in 2018. 2018. Firstly, I would like to start with the urgency of machine learning. As we know that machine learning is transforming every part of our world, from the emails to genes, for the emails we send to genes we study. For example, in healthcare, AI detects disease earlier, accelerates drug discovery, and enable personalized treatment through data-driven insights. Crucially, in agriculture, for example, AI enables precision farming, as mentioned by Professor Seishi, and early crop disease detection, smart irrigation, and better yield prediction, helping build more sustainable and resilient food systems. So AI, including machine learning and or deep learning, is not just technologies. AI is the driving force of innovation, seamlessly integrating into our daily lives as intelligent system operating real time everywhere. So how actually machine learning works? Machine learning actually is a process that enables computer to learn from data and improve over time without being explicitly programmed. So usually we start with gathering data collected from, for example, from sensors, image, text, or user interactions. And next, we train the model using this data, the training data, so it can recognize or find the interesting patterns and relationships. Once trained, the model is packaged into a deployable form and validated to ensure it performs accurately and reliably on new unseen data. After validation, the model is deployed into real world. We then monitor its performance continuously because real world data can change over time. If performance drop, for example, or new data becomes available, the model goes through retraining again, allowing it to adapt and stay accurate. So in essence, machine learning system analyze pattern, handle complex and high-dimensional data, and detect nonlinear relationship that traditional method might miss. Okay, I would like to focus on the genome-wide association studies, or we call it as GWAS. GWAS actually help us understand how small changes in our DNA can influence phenotypic traits, or in this case, influence the disease. In our genome, there are millions of points where a single base pair, such as A as adenine, T thymine, C cytosine, or G guanine, can vary among individuals. These are called SNPs, or single nucleotide polymorphisms. Most SNP actually have no effect, but some occur more frequently in people with a particular phenotype compared to those without it. For example, in this case, SNPs 1 and 2 show no association with the disease, but the SNP 3 appears much more often in individuals with the disease, suggesting that it may mark a region of the genome involved in disease or specific phenotypes. The SNP act as a genetic markers. So if we, for example, collect data from next generation sequencing, so we will get a whole genome sequencing, and we can get a genomics marker, call it SNP. SNP helping researchers locate genes and biological pathways that contribute to the disease or specific phenotypes. How actually GWAS in plants? GWAS in plant help us understand how a SNP can influence a plant's observable traits or phenotypes. The process begins with genotyping, where DNA from different plants is sequenced and analyzed to identify SNPs. This variant may include insertion, deletion, or mutation, or we call it as SNPs. At the same time, phenotyping is conducted to measure traits such as heat, leaf, size, flower color, fruit yield, or biomass, and so on. And advanced imaging and data management, such explained by Professor Seishi, are often used to collect this phenotypic data accurately and efficiently. In the association analysis, genotypic and phenotypic data are integrated using statistical or machine learning is GWAS methods. We identify significant genotype-phenotype association. So the objective is how to find the association between genotype and phenotypes, marker trait associations, or we call it MTA. From this, we can locate quantitative traitology, or QTL, genomic regions that influence key agricultural traits. Through this approach, GWAS provides a powerful framework to uncover the genetic basis of complex traits in plants, guiding crop improvement and precision breeding effort in agriculture. So why there is the question why traditional method miss the mark in large genetic studies? First, they rely on the traditional method, really on linear assumptions. The models expect simple, just one-to-one relationship between genetic markers and traits, but in reality, biological systems involve complex nonlinear interaction among genes. We call it as epistasis, and traditional models fail to capture the epistasis interaction between genes. So the second, there is a single SNP focus. Conventional GWAS typically analyze one SNP at a time, so which means they can miss the combined effect of multiple SNPs that may jointly influence a trait. Third, we face the high-dimensional challenge. Modern data can contain millions of SNPs, but only a limited number of sample or SNP that is very important. So modern data sets, maybe we have, for example, millions of SNP, but only from the a limited number of samples. So there is an imbalance between variable of feature and the number of samples. This is actually in computer science terms often called as the course of dimensionality. So make it difficult for traditional statistical approach to handle this problem. So finally, population stratification can lead to false positives. Differences in ancestry among individuals may appear as genetic association with traits when in fact they are simply due to population structure. This limitation highlight why integrating advanced approaches like machine learning and system-level analysis has become essential to capture the true complexity of genetic variation, especially in plant and other diverse organisms. So, just want to talk more detail about a machine learning in GWAS study. First, we have a regression model. This is actually used when we want to predict continuous outcome, for example, for knowing the association between genomics and phenomics. And the second is classification models are useful for predicting categorical outcome, such as, for example, classifying plants or traits into distinct groups. For instance, disease resistant versus susceptible varieties. And the third is assembly learning. This is actually combines multiple models to improve prediction accuracy. This approach is particularly powerful for identifying important features, such as a key SNP gene, because, for example, if you use a random forest actually develop based on the decision tree. So we can identify the important features that affect the decision of the machine learning. And also, assembly learning is very powerful for handling nonlinear interaction between variables, and also effective in integrating multiple data sets, which is common in plant genomics. And finally, we can use deep learning or neural networks that can capture highly complex and nonlinear relationship that traditional model may be miss or cannot do to find the relationship. They also enable automated phenotyping and feature learning and helping bridge genomics and phenomic data for deeper biological insight. So this machine learning approach makes GWAS more robust and insightful, allowing researchers to better understand the genetic architecture of complex traits and accelerate crop improvement compared to the traditional ones. Okay. This is actually our study. Our study show an example of how machine learning can optimize SNP selection using variable ranking and advanced selection techniques. In a typical genome-wide association study, a single dataset can contain hundreds of thousands of SNP, while an exhaustive search can identify. So we can use the exhaustive search to identify the best combination of SNP associated with a trait, but it's very computational intensive. So to address this challenge, machine learning offers more efficient SNP selection method. So we use the approach using the combination approach using machine learning random forest and SFFS that can rank variable based on their importance and filter out uninformative SNP early in the process. So by doing this, we can reduce computational load very significant. And also improve accuracy by focusing on SNP that are truly associated with the traits of interest rather than random noise. And this is actually more detail of our approach. This is the workflow for optimizing SNP selection using a combination of feature ranking and advanced selection techniques. We start with genomic and phenotype data as input. We have two data sets we test here. one with 1,000 SNP without epistasis, and another with 10,000 SNP, including epistasis interactions. The first step is SNP ranking, which acts as a feature filtering process to reduce the search space. In this step, SNP are ranked based on their importance or correlation with the three ranking methods we applied. So we use the three methods, the CAR score, which measures statistical correlation, the I-score, which reflect influence-based ranking, and random forest machine learning approach that ranks SNP by predictive power. The result of this step is a subset of top-ranked SNPs. And the next step is SNP selection is performed using the sequential forward floating selection, or SFFS. This algorithm iteratively add or remove SNP to identify the optimal subset that best predicts the target trait. So then we use support vector regression model is to test using two kernels, the radial basis function and the basal kernel. So the SVR actually used for predict the score of association between genotype and phenotypes. Finally, the selected SNP are validated through model evaluation using R-square to measure how well the SNP explain trait variations, and mean square error to assess prediction accuracy. The final output is a set of validated SNP with both strong statistical and predictive value. Okay. So our ranking, this is actually the result. Our ranking experiment showed that the random forest algorithm significantly outperformed traditional method like the CAR score and I-score, particularly in efficiency, reducing the search space for complex high-dimensional data. So this is proof that machine learning, actually in this case, is better than traditional statistical approach. For the selection state, the SFS method, particularly when using adjusted R-square and mean square error as selection criteria, demonstrated superior performance compared to the previous one. So what actually the key insight from this research? The initial step of reducing the search space with variable ranking is very crucial. So this step, combined with our advanced selection techniques, significantly improve the result, especially for phenotypes involving complex epistasis. So while also dramatically shortening computation time. Now I would like to talk about the future direction for machine learning in plant GWAS. To truly maximize the impact of machine learning in plant genomics, we must focus our effort on two critical areas. The first, we need to focus on improving machine learning model themselves. And there are 4 key challenges for improving machine learning models. First, we need to develop robust validation method. Our model must perform reliably, not just on local data, but on large, diverse, and independent data sets from various breeding programs and environments. So data set is very important, as Professor Seishi explained in their presentation. And we must work on creating automated and scalable processing workflow for GWAS data. And the third, a major challenge in developing machine learning approach that can properly account for genetic diversity across population. Current models often struggle to generalize finding between distinct genetic backgrounds. And finally, we must explore techniques to make our machine learning models more interpretable. This is very important because geneticists is not enough to have a good prediction. We need to understand whether the model made that prediction to gain meaningful genetic insights. Sorry. The second major direction is how to integrate omics data for post-GWAS analysis. We know that the biological system is very complex and single SNPs only tell part of the story. So our focus here is on developing machine learning approach to integrate SNP with other omics data such as transcriptomics, proteomics, and metabolomics as illustrated in the top right of the figure. This comprehensive analysis will allow us to move beyond simple association. So we will get a more complex association between all the data, omics, and also from genomics, transcriptomics, proteomics to phenomics. And the ultimate goal is to gain deeper insight into the molecular mechanism and the complex phenotypic network that underlie important plant traits. So by combining genomic, molecular, and phenotypic data, we can build a holistic view of the plant's biology. So this is very important. And what actually machine learning for precision agriculture. So moving from research theory to real world application, this actually illustrates how machine learning is becoming a cornerstone of precision agriculture. The integration of machine learning with genomics, particularly GWAS, provides four crucial benefits. The first is machine learning enabled GWAS help us to identify the key genes that drive crucial trait association. This is vital for improving, for example, growth and heat tolerance, enhancing nutrient use efficiency, and or bolstering disease resistance in our crops. And by providing highly accurate insight, machine learning enables data-driven breeding and facilitates site-specific crop management. So this means tailoring our action precisely to the needs of a particular field or microenvironments. And then machine learning strongly support predictive phenotyping. It allows us to model how plants will respond to complex, changing environmental conditions, moving beyond simple observation to accurate forecasting. This diagram on the right summarizes this integration, showing how data collected, for example, from satellites and drones, like video and image data, are combined with underlying genomics and phenotypic data to inform every aspect of crop improvement and management, from sensing and soil management to yield prediction. So machine learning is the engine driving this data to decision pipeline in modern agriculture. Our final point focus on the critical need for collaboration, as mentioned by Professor Seishi, that we need a collaboration, specifically fostering interdisciplinary work between machine learning and statistical genetics. The challenges such as complex trait analysis to integrating diverse omics data cannot be solved by only one discipline. We must build framework for cross-disciplinary research to effectively address the growing complexity of GWAS by creating common language and standardized data structure that both computational scientists and biologists can use. So the combination between the computer scientists and biologists is very important. So we should build the common language and standardized data structure. The goal is to actively merge cutting-edge machine learning innovation with time-test statistical genetic insight to solve the grand challenge in plant biology. For example, machine learning can identify subtle pattern, but statistical genetic provide the hypothesis, rigorous hypothesis testing, and population structure awareness needed for biological validity. This is actually the diagram illustrates the complexity of the research area. It involves plants, the environment, pathogens, all interacting through molecular network, disease, and agriculture practices. Successfully mapping the genetics of this entire system requires a truly integrated approach. So by breaking down disciplinary silos, we can accelerate discovery and translate genomic knowledge into meaningful agriculture solutions. This is my talk, actually. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Professor Wisnu Ananta Kusuma. Hatur nuhun pisan, Prof, for that very informative and thought-provoking presentation. It's also very impressive, ladies and gentlemen, to see on how machine learning has really taken crucial role on the latest development instead of use it of the traditional statistical approach, as clearly mentioned by Professor Wisnu Ananta. All right, now we will move to the Q&A session. And as what we have together witnessed on how the latest development of the impressive implementation of the digital advanced technology has been supporting us, especially in Phenomics. So now both experts have shared, cleared it out to all of us. Should you have any question, of course, to all participants, please type your question in the chat box or use the raise hand feature. We will select the several questions to be addressed directly by our speaker. Prof. Seishi Ninomiya already here. Prof. Ananta still around. All right. So, committee, please share us the question we received in the chat box. First question, please. Next. All right. Let me read this out for everybody benefit. From Trias Sitarasmi, affiliation IPB students or BRIN, the research center in Indonesia, to Professor Seishi Ninomiya. I'm doing research on digital phenotyping by using DSLR camera and UAV for rice tolerance to iron toxicity, focusing on yield, canopy, and leaves symptom. The question is how to get high accuracy of prediction using deep learning model. Please, Prof. Seishi.
Okay. Well, this question is kind of very general. So, I mean, I do not know what kind of situation he or she is doing his research, but I mean, well, when we speak about the accuracy, there are two aspects. Okay, very accurate to someone's own data set. Or another thing, how general the model is to the whole data set, you know. Actually, when we like to use the approach, yeah, the second is more important. Generality of the model is more important. So if this questioner asking about that kind of accuracy, then only thing I can say is that to collect more data to make it from the different locations, different situations. And also, you know, something very difficult in Phenomics is that, I mean, observation and the result depends on, say, when we collect data is very important because, you know, DNA is a static, but actually DNA expression is not a static, very dynamic. So depending on the time of the observation, everything changes. So that makes everything very, very difficult, actually. And we have lots of discussions. But still, so he wants to tolerant to iron toxicity. This may be quite static, I may, but still there's some interaction of the plant physiological situation. So also the time of the observation must be very important. And sorry, it's not easy to answer this question. I kind of very general. So okay, maybe I don't. Okay, so the questioner cannot speak, and just they can type the questions, right?
They can also speak. So allow me to give chance for Trias Sitarasmi. Are you still around? Would you like to share the question directly to Prof. Seishi Ninomiya? Are you available, Mbak Trias? Should you also available to speak or to share in Bahasa Indonesia, then we can do something about it if you are available.
Yeah, actually, the lot.
Unfortunately, she's not around, Prof. So meaning she's not able to speak for now. Please go ahead, Prof. Seishi.
Yeah, actually, there are lots of similar studies. So yeah, he can you know make some survey on the papers published before, I think. Anyway, the time of the observation is very important. Also, the kind of observation by UAV can easily influenced by the solar radiation, the sun altitude, etc. Many and one of the functions when we conduct statistical research is outdoor. So must be very careful about the conditions. I don't know if I have
Well
answered
noted,
the
Prof.
question. Yeah,
Seishi.
yeah, okay.
Yeah, okay. Thank you so much for the answer, Prof. Seishi. I hope that will more than sufficient to answer this question. So, Pak Wisnu, would you like to add something on that question?
Sorry. Okay. Yes. I think I agree with Professor Seishi that uh, to get the high accuracy of predictions using deep learning model, actually we need the data sets, and the data set should be representing the many conditions of the for example the genomics and also phenomics. So we should have uh, the good quality of data sets. This is actually very important. If for example, if we don't have the the good quality of data set, or just few of data sets, maybe we can use the many techniques in computer science how to uh, I forget the terms, how to make the like mm, to to increase the size of the data using the simulated or simulated data set, for example, like that. But this is actually will affect of the our accuracy. So the data sets is very important, and also the architecture of the deep learning also very important. Maybe then maybe I just would add like this. This is my opinions.
Thank you, Prof. Wisnu, for the additional valuable insight and information. So now let's go to the next slide, the second question. All right, from Nurfianto, affiliation Malang. Malang is the name of the city in East Java, Professor Seishi Ninomiya. To Prof. Wisnu, the first question, he has two questions. The first question is machine learning, or are machine learning and artificial intelligence development for GWAS and genomic already patented and used by plant breeders? And the next question, the second question is, how is the competitive advantage of machine learning that is developed with many new tools and analysis from biotechnology industry such as Illumina, bioinformatics, analysis, etc., etc. Prof. Wisnu, the time is yours.
Okay. This maybe I didn't do the survey for that, but I think, for my opinion, machine learning method or for genomics and they're used in breeding actually already patented by a large company and widely used in commercial plant breeding by plants. But at the same time, many powerful ML machine learning tools remain open and are commonly adopted by academic breeders. So uh, yes, maybe we can still use the as academic breeders, we can use the powerful tools that open access, for example, like that. And the second question, can you repeat the second question, Mr.
Oh, sure. But can you see also the screen, because we also
Okay,
presenting it
okay.
on the screen? Okay,
Okay, how is the
how is
competitive?
the
Okay.
Yes, please. Yeah,
Yes,
right.
actually, as I already explained before in my presentation that uh, using machine learning approach, actually we will get a faster and more accurate selection uh, genomics marker samples. Yes, uh, compared to the traditional statistics. If, for example, dealing with the nonlinear problems and also the high dimensional data. For example, we have a many 10,000 or a million SNP, for example, like that. And also the advantage is scale and integration of heterogeneous data So combining high throughput sequencing, for example, from Illumina, and also remote sensing, Phenomics, environmental, and other data, we can get a more accurate analysis to make association between kinetics and genotypes and phenotypes. So using machine learning, we can scale and scale and make an integration of heterogeneous data. We can say that this is more multimodal problem that cannot be solved easily by using statistical a traditional statistical approach. And also we can automation and decision support. So machine learning systems can make a recommendations, for example, which seed to plant, where management advice, and also like a decision support system. We can develop the decision support system based on the machine learning. Like that. This is maybe my questions. Sorry, my answer.
Thank you so much, Prof. Wisnu. I'm now giving opportunities for Prof. Seishi Ninomiya. Would you like to add on what Prof. Wisnu has shared to us, Prof?
Oh, yes, as he mentioned, there are several, open tools which are available for everyone. And I know that the performance not worse than, commercially available tools, so they can use them. And the most important thing is that, just going back to the first question to me, I mean, if data set is small, I mean, you cannot apply that. You cannot develop anything. It is, in the case that the data is small from your own field, you can develop a very good site-specific model to just for yourself, but it is usually useless. So, I mean, in addition, even for the GWAS case, so kind of big variation data set is necessary for them to have a good estimation, considering background of the data set, because background is too different. The result, changes. I don't think I have enough time to explain about the background. And for example, if you that, the indica and the japonica for rice, and if you mix those things all together, maybe that kind of background can influence the result of the GWAS. So you have to be very, very careful about how to select your data set and the scale of the data. Very important. Which is more important than which tool you use, I think. Sorry, I haven't answered the question, but just I'd like to emphasize the most important things here when you do these kind of things. Thank you.
Thank you, Prof. Seishi Ninomiya. I believe everybody 100% agree on that. Tools are available, information are available, but on how we convert the information into data, on how we collect the data, we utilize the correct and appropriate data will be the more important things. Thank you so much, Prof. Wisnu and Prof. Seishi. Now let's move to the third question. The third question is from Zaki Abdul from IPB to Prof. Seishi Ninomiya and to Prof. Wisnu Ananta. In Indonesia, we have many corn and paddy hybrid varieties, and it takes much time to develop it. One of the most difficulties in developing hybrid varieties is in selecting suitable parents. The question is, how machine learning and artificial intelligence can help to foster the development of hybrid varieties? I shall give the opportunity to Prof. Seishi Ninomiya first. Please, Prof.
Yeah, okay. This is a very, very good question. And this is one of the targets of modern plant phenomics. I mentioned that the final kind of ultimate goal of phenomics is to develop a model to explain phenotype by genotype and the environmental condition and management, such as the fertilizer levels. Okay. Once we have a model, we can conduct a so-called design breeding, which means that we can select the parents most optimal way to predicting the performance of the hybrid, for example. So, well, this case is again a little bit, I mean, simple. But anyway, machine learning or deep learning, whatever, kind of so-called AI approach will help this a lot. But I think this is one of the most hot topic of the scientists in this field now. Many people are try to develop a system to design cultivars digitally and try to realize that one based on this design. This is not just for the hybrid, but for any cultivars. We should be able to take this away in near future. Thank you.
Thank you, Prof. Seishi. Now I shall give a chance for Prof. Wisnu Ananta to add on that.
Okay. Yes, actually the process maybe already answered the question, but I would just want to add the emphasize that this is in from the perspective of computer scientists that the objectives of the machine learning in this case actually is to reduce the search space. You know, the search space. For example, if we have the such number the large number of SNPs, so we can select the few SNPs that have the high association with phenotypes. But if, as explained by Professor Seishi, if we don't have the good data sets, for example, the good phenotypes and so on, especially for development of hybrid varieties, so maybe the accuracy of the making association between phenotype and phenotype is also low. But the most important is using machine learning, we can reduce the combinations, the use of combinations of SNPs, especially related to the epistasis problem. Maybe this is just my answer. Thank you.
Thank you so much, Prof. Wisnu Ananta. All right. Ladies and gentlemen, before we go to the next written question, I would like to deliver an opportunity for participants who would like to share the question directly or verbally. Can I see a raised hand on this session? Anybody, participant, who would like to share his question directly, verbally to the speakers?
Yes, please.
Oh, Miss Sausan. Go ahead, Miss Sausan. Please share your name and your background, your institution.
Yeah. Good morning, everyone. Thank you very much for the opportunity. My name is Sausan. I currently work at Ispasid Indonesia. Hello, Pak Wisnu. It's really nice meeting you again. Good morning.
Hello, Sausan.
Yeah, so it was quite tricky for me to understand the concept of SNP ranking. I would like to ask if it's possible for you to explain more, because you claim that SNP ranking is better for data with for phenotype with epistasis. As far as I know, that SNP ranking ranks individual SNPs, but with epistasis we work with combination of SNPs. That's it.
Yes, thank you. This is very difficult question, Sausan. Actually, maybe I just want to answer about this. It's different between the SNPs ranking and epistasis also. In our study, we use the random forest as the one of the machine learning method to make a ranking. So, as we know that SNP in this case is as features. So if we have, for example, 10,000 SNPs, so we have the 10,000 features. And by using random forest, we can then like doing feature selections automatically and can calculate the importance scores of each features. So by using importance scores, then we can rank the SNP from the high importance score until to the lowest importance score. This is actually how we did with this study. Is this clear? Maybe hopefully answer the question. Huh?
Yeah.
Maybe
It's a
if you want
question.
more detail, you can come to the Tropical Biopharmaca Research Center in Bogor, and we can
Sure.
discuss.
Thank you for the invitation.
yeah,
I
yeah.
will plan a visit to IPB.
Yeah.
Thank you, Pak.
Thank you.
Yeah, thank you for the question, Ms. Sausan, as well as what Prof. Wisnu Ananta has mentioned. You are very welcome to visit us in IPB. All right, thank you. So now let's move to the next written question. Participant, ladies and gentlemen, we still have two written question. Committee, please show the fourth question, the fourth written question. All right, from Bismira Gandhi Ahmad. Affiliation from PTCRA is an enterprise, private sectors, Cahaya Rapi Abadi, to Prof. Seishi Ninomiya. What wavelengths or spectral bands are used to monitor plants' condition, and what formulations or indices are applied? Prof. Seishi Ninomiya.
Yeah. Okay. So again, this is a bit too general question to me because it depends on what we like to observe, what kind of trait we like to, evaluate. So, well, we used every combination of the wavelength, but for example, the simplest one, for example, I mentioned about the detection of rice flowering. In that case, we used only RGB. And even though the various light condition, our approach worked very fine. But for example, if you like to monitor photosynthesis, then you have to use very special wavelength. And also, as you know, now hyperspectral imaginary systems are available, which can provide us maybe 300 to 400 different wavelengths in just in one sensor. In that case, most important thing is to find out most optimal combination of wavelength. And this part is again, it's kind of very, very hot spot of our phenotypic researches nowadays. So it's not easy to answer to your question, but depends on what you like to monitor. I mean, architectural trait, usually visible, RGB image is sufficient because it can observe as human can do. But some easier conditions, like water use efficiency or radiation use efficiency, etc., cannot be directly measured by visible sensors. Then we have to use other things. Have I answered to your question? Still, yeah, if you can mention some specific trait, then I may be able to add some information for you. Thank you.
Thank you, Prof. Seishi. So, Bismira Gandhi Ahmad, are you available to share what Prof. Seishi has requested, the example? And maybe it's not available to share it out now. Oh, he's in writing. Okay, thanks for the explain and answer. So that's a thank you confirmation from Bismira Gandhi from the gentleman who asked the question, Prof.
Okay,
So I
thank you
think the
very much.
explanation from you are clear enough. Yeah, Prof. Wisnu, would you like to add something before we go to the last question?
I think it's enough. Thank you.
All right,
No,
thank
no
you.
problem.
The next question, the fifth question. Oh yeah, participant, ladies and gentlemen, so sorry, time is limited for us, but of course, all the received question, because Prof. Seishi and Prof. Wisnu, we still have many written question. We will be sharing it out to you, sharing them up to you, to Prof. Seishi and Prof. Wisnu, and please answer in written so that we can distribute to the person who raised the question. All right, the fifth question is going to be the last question from Jasmine Tasmara. Affiliation is West Indonesia. This is also a private sector enterprise. To Prof. Seishi Ninomiya. Professor, we know that developing a digital plant phenotyping systems requires a large amount of data, especially for deep learning approaches. I like to ask, what will be the best way to acquire high quality and accurate data for these purposes? And when collecting image data, is it better for the data set to be uniform, for example, with consistent lighting and background, or more diverse to help the model generalize better? Please, Prof. Seishi.
Yeah, okay, this is again a very good question. And well, okay, for first, I would like to answer to the second part about the condition of, I mean, again, it depends on how you like to use your approach, how you like to apply approach. Depends on what you like to do. But in general, we like to use the AI system for monitoring the outfield to help the farmers or help the breeding field, etc. So the system must be robust under the natural environmental conditions, which means that you have to collect data from outdoor. Of course, that makes the things more difficult, but very simple model developed in indoor. Of course, you can provide several different combination of environmental conditions, but it takes much time, and I don't think it's a good idea. Better thing is to do it outside and try to make it robust under the natural conditions. That is what our team has been doing outside. And when we started this approach, people didn't believe, because most of the phenotyping studies have been being conducted indoor in European countries, providing a huge facility or something else. But we thought that about 20 years ago, we thought that that approach won't work outside and practically. So that's the reason why we took the more difficult way to provide some systems. So I would like to, and again, depends on what you like to do, how you like to use your system for the monitoring. But if you like to do it, like use for the outside, yeah, the general answer is to do it outside. Of course, there are lots of things which are very difficult. So very, very beginning, maybe you can try it under the uniform condition. But once you have some more information, then you should try do it in outside. What is the first part of the question? What's the,
Yes,
okay.
Prof.
Yes,
I would say yes.
Yeah. It's also, I mean, I think I have already answered to the question as well for the first part. Yeah.
Yes, Prof. Yes, you have. You've done it. All right. So I would like to give an opportunity for Prof. Wisnu Ananta. Would you like to add on this question?
Thanks. No, maybe. Yes.
All right.
I don't have
So thank
experience.
you.
Okay.
No, well, thank you, Prof. Wisnu Ananta. So, ladies and gentlemen, participants, that's going to be the end of the question. We have 5 questions in written and 1 question in verbal. But of course, if you like to be able to meet our 2 remarkable presenter this morning, Prof. Seishi, Ninomiya, and Prof. Wisnu Ananta, we welcome you to the APFITA 2025 in Bogor, which is going to be held on November 17 up to November 19. So please make sure that you register so that if you have any further follow-up question, you can go directly tunnel to both speakers. Now we will move to the concluding remarks session. So it's again honor to invite Professor Yandra Arkeman, a professor of agro-industrial engineering and computer science, to share his remarks and scientific reflection on today's presentation, particularly from the perspective of system engineering, AI, and digital integration in agriculture. Prof. Yandra, are you with us? Oh, he's already with us. Okay. All right, Prof. The time is yours, Prof. The screen.
Okay. Kameranya belum hidup. Ya, I just want to turn kamera. Oh ya,
Yeah,
ya.
we can see you, Prof.
Okay, good. Okay, I am very happy now to sit here. Just have followed the very, very great, presentation from Professor Seishi Ninomiya and also Professor Wisnu. This is my concluding remark that this webinar is actually a crucial step toward bridging advanced and scientific research and also practical agricultural application. Your engagement and curiosity will be key drivers in advancing plant phenomics in IPB University and also in the world actually to benefit global food system. Yeah, our global food system will be take benefit for this seminar, and also environmental resilience. Let us embrace this opportunity to learn, connect, and innovate together. And also don't forget, Pak Verry and team should also follow up with the real collaboration between our university, IPB University, and also Tokyo University, and maybe other universities. Again, thank you very much for your participation, especially Professor Seishi and Professor Wisnu, and also our great moderator, Pak Verry Surya Hendrawan. I close this webinar with thank you very much. Alhamdulillah Rabbil Alamin. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam, Prof. Yandra. Ladies and gentlemen, we not yet finished. We still have a session. So that's a concluding remark from Prof. Yandra Arkeman. Thank you, Prof. Yandra. Together, this insight, what we have seen today, experienced today, shows that plant phenomics is not only a scientific field, but a strategic pathway toward building a resilient, data-driven, and sustainable agricultural future. So we have reached the final part of my turn today in this Today International webinar. I would like to return it back to the Master of Ceremony. Apologize for any mistake that I made. Mohon maaf untuk segala kesalahan. Izin saya tutup. Wallahu muwaffiq ila aqwamit thoriq. Nasrun minallah fatun qarib babasirul mu'minin. Assalamualaikum warahmatullahi wabarakatuh. Master of Ceremony, the stage and place is yours.
Warahmatullahi wabarakatuh.
Many thanks to the moderator.
It's frozen. Master of Ceremony is frozen. All right, committee, could you please move to next screen, which is
Mr. Verry
the
Hendrawan
token.
and our two speakers for an interactive and insightful session. So
Please wait a
Go ahead.
minute, Pak Verry.
Yes, ini sertifikat seems that our MC is in trouble with the signal. So I would like to just make it quick. Mbak Nanda, are you there?
Yes, I'm here.
Okay, please proceed, Mbak.
thank you for your information. We hope we'll participate in the conference. It's time to appreciate the contribution of our practice. We will continue with the symbolic certificate presentation session to the moderator and speaker. Professor Yandra gave a certificate to speaker a symbolic certification. The professor, welcome.
Okay, thank you very much. First, I would like to give the certificate for Professor Seishi Ninomiya. Yeah, I deliver the certificate online, so please accept the certificate, Professor Seishi Ninomiya.
Thank you very much.
Thank you very much. You're welcome.
Also, I put my presentation PDF on the on chatbot so people can get it from there.
Okay, thank you very much. Next certificate will be for Professor Wisnu Ananta Kusuma.
Thank you very much, Prof. Yandra.
So I give the certificate to you as token of I give the certificate to you as token of appreciation for Professor Wisnu. Please accept our token of appreciation and certificate.
Thank you very much, Prof. Yandra.
Yeah, you're welcome. Last but not least, I would like also to give the certificate to our great and full of spirit moderator, Pak Verry Surya Hendrawan. Please
Thank you
accept
very
our
much.
certificate, Moderator.
Thank you very much, Prof. Yandra. Really appreciate for the opportunity given for me to be a moderator on this great and insightful webinar. Thank you so much.
You're welcome.
Thank you, Professor Yandra, for certificate, for our moderator and our aspiring speaker for today. So very, very amazing. It's time to appreciate. With the completion and certificate presentation session, the entire series is International Webinar Pre-APFITA Series Chapter 3 event today concludes. We extend our deepest gratitude for your active participation. On behalf of entire committee, we apologize for any shortcoming during the event. I am Nanda Ibrani. As a master of ceremony, I apologize for any mistakes in international or not. See you in the next webinar. Good afternoon and best wishes for us. Wassalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Prof. Seishi, see you in Bogor, Prof.
Okay,
Looking
see you
forward to
then.
see you in Bogor.
See you,
Yes.
Prof. Seishi.
Yes, okay, very good. Thank you very much.
Thank you very much.
Terima
Arigatou
kasih semuanya.
gozaimasu.
Assalamualaikum
Hatur nuhun, Pak Wisnu. Hatur nuhun.
Sama-sama
Sampai
ya.
ketemu, Pak.
Amin.
Hatur nuhun, hatur
Assalamualaikum.
nuhun.
Mangga.
Prof. Yandra. Assalamualaikum warahmatullahi wabarakatuh. Masih, Prof. Masih, Prof. Yandra.
Pak Bayu, terima kasih Pak Bayu,
Prof.
Maliana.
Ya, makasih
Ya,
Pak
Mohon
Ferry
hadir juga Pak
Prof. Seishi,
nanti.
insya Allah
Pak
nanti
Bayu, hatur
ketemu di
nuhun, Pak
Bogor
Bayu.
lagi Pak. Ya, Pak Wisnu, Pak Ferry.
Pak Bayu ada.
Siap, Pak Bayu.
Mohon
Hatur
juga
nuhun,
nanti
Pak
semua
Bayu.
hadir di APFITA.
Terima kasih semua, insya Allah nanti dari Pusdatin hadir juga Pak Yandra. Izin
Siap,
live ya, Pak.
alhamdulillah.
Assalamualaikum.
Izin live.
Siap,
Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. Mangga, mangga. Nuhun, nuhun.
Berapa orang peserta?